Welcome back, everyone. We're about to take a deep dive into the world of microbial growth. We've got some really cool research lined up.
Yeah.
All about how these tiny organisms just multiply and thrive in conditions you wouldn't believe. Think boiling hot springs,
Oh, wow.
solid rock, even those leftovers in the back of your fridge.
It's incredible, right? These tiny organisms have adapted to survive in almost any environment imaginable.
Right, and that's what we're going to be exploring today.
Absolutely.
How do bacteria reproduce? What environmental factors influence that growth?
Right.
And how do scientists even begin to measure how many bacteria are in a sample?
Well, we'll be diving into all of that, and we're even going to be exploring the fascinating world of biofilms.
Oh, yeah.
These microbial cities where bacteria team up to create these incredible structures.
It's amazing. Okay, so first things first.
Yeah.
How do these microscopic ninjas actually multiply?
So most bacteria reproduce through a process called binary fission. And essentially, one cell just splits into two identical daughter cells.
So it's like a microscopic cloning machine.
Exactly.
And I'm guessing that happens pretty quickly.
Oh, absolutely. The time it takes for a bacterial population to double is called the generation time.
Okay.
And for some bacteria, this can be as short as 20 minutes.
Wow.
Under ideal conditions.
No wonder that food left out on the counter can spoil so quickly.
Yeah, that's a perfect example. Now, in a closed system, like a container of food, bacterial growth follows a predictable pattern with 4 distinct phases. Lag phase, log phase, stationary phase, and then the death phase.
So during that log phase, they're multiplying like crazy, right?
Exactly. That's when their growth is exponential.
Got it.
And they're just gobbling up all those nutrients and dividing as fast as they can.
Makes sense. So let's say a scientist wants to figure out just how many bacteria are in a sample.
Okay.
Maybe it's a contaminated water source or a food product. How do they actually count these tiny critters?
So there are actually several clever methods, both direct and indirect.
Okay.
Direct methods involve actually counting individual bacterial cells or colonies.
Okay.
So for example, we have the pour plate and spread plate techniques.
Okay.
These allow us to isolate and count colonies on a nutrient agar plate.
I see.
Each colony arises from a single bacterium. So by counting the colonies, we can kind of back calculate and estimate the original number of bacteria in that sample.
So it's like a microscopic census?
Precisely.
Okay, that's cool.
Yeah.
What about membrane filtration?
Ah, yes. Membrane filtration, that's another direct method.
Okay.
Where we basically use a tiny sieve to catch bacteria from a liquid sample.
Got it.
And then we place the filter on a nutrient agar plate, and the bacteria grow into colonies that we can then count.
So clever. What about those indirect methods you mentioned?
Right. So indirect methods rely on measuring something that correlates with bacterial growth rather than counting individual cells. One common method is measuring turbidity.
Turbidity.
Yeah, or the cloudiness of a liquid culture.
Okay.
So the more bacteria present, the cloudier the culture becomes.
Ah, that makes sense.
It's like judging the density of a crowd based on how hard it is to see through it, right? And we can even use a spectrophotometer to actually measure the absorbance of light by that culture, giving us a precise measurement of turbidity.
Oh, wow. Pretty high tech. Are there any other indirect methods?
Yes. We can also monitor bacterial metabolic activity.
Oh, okay.
As bacteria grow, they consume nutrients and produce waste products.
Right.
And by measuring these changes, we can estimate their numbers.
So it's kind of like tracking their breathing and eating to get a sense of how many are there.
Exactly. A great analogy.
Now, we've been talking about binary fission.
Right.
But is that the only way bacteria reproduce?
So while it's the most common, not the only method, some bacteria can divide by fragmentation, where a long filament breaks apart into smaller pieces, and each piece can grow into a new individual.
Interesting.
Others, like certain types of yeast, use budding, where a small bud kind of grows off the parent cell and eventually detaches to form a new cell.
So it's not just about splitting in... Bacteria have a few reproductive tricks up their sleeve.
They do.
Regardless of how they reproduce, though, their growth is heavily influenced by their environment, right?
Absolutely. Environmental factors play a crucial role in determining how quickly bacteria grow and even whether they can grow at all. One of the most important factors is oxygen.
Yeah. Okay, let's talk about oxygen.
Okay.
We need it to survive, but some bacteria I know they actually find it toxic. It's kind of mind-boggling.
It is. It all comes down to their metabolic pathways. Bacteria can be categorized based on their oxygen requirements.
Yeah.
So we have obligate aerobes like us who need oxygen for survival.
Right.
They use aerobic respiration, which is a process that uses oxygen as a final electron acceptor to generate energy.
So they're breathing oxygen, just like we do. Okay, what about those bacteria that can't handle oxygen at all?
So those are called obligate anaerobes.
Yeah.
Oxygen is actually poisonous to them.
Wow.
They rely on processes like fermentation or anaerobic respiration, which use other molecules as final electron acceptors.
Got it.
For example, some bacteria use sulfate or nitrate instead of oxygen.
So there are bacteria out there that would literally suffocate in the air we breathe.
Precisely.
Wow, that's wild.
Okay.
And then you mentioned facultative anaerobes.
Right. Those are the flexible ones.
Okay.
They can grow with or without oxygen, switching their metabolic pathways depending on what's available.
So it's like having a backup generator.
Exactly.
Okay. I've also heard of aerotolerant and microaerophiles. What about those categories?
Right. So aerotolerant anaerobes, they don't use oxygen for respiration, but they can tolerate its presence.
Okay.
Microaerophiles, on the other hand, need oxygen, but at much lower concentrations than what we find in the atmosphere. They're a bit picky about their oxygen levels.
So it's not just about needing or not needing oxygen. It's about finding that sweet spot for each species.
Exactly.
Okay. And speaking of finding that sweet spot, another crucial factor for bacterial growth is pH, right?
Absolutely.
I mean, our stomachs are pretty acidic. Are there bacteria that can handle that kind of environment?
You bet. Most bacteria prefer a neutral pH and are called neutrophiles.
Okay.
But there are also acidophiles, which thrive in acidic environments, and alkalophiles, which prefer alkaline conditions.
So those bacteria in our stomachs are like extreme sports enthusiasts, loving that acidic environment.
A great analogy. And remember, pH can have a huge impact on bacterial growth.
Right.
Too acidic or too alkaline, and their enzymes can't function properly, and that can slow down or even stop growth.
That's all about finding that Goldilocks zone, right?
Yeah, exactly.
Besides oxygen and pH, what about temperature? I mean, from the frozen tundra to boiling hot springs, bacteria seem to have conquered it all.
Indeed. We classify bacteria based on their preferred temperature ranges. So we have the psychrophiles. They're the cold-loving champions, thriving in temperatures from 0 to 15 degrees Celsius.
So that's why our leftovers eventually go bad, even in the fridge.
Exactly. Those are the soaker troughs at work.
Wow.
Then we have the mesophiles.
Okay.
The moderate temperature lovers, growing best between 20 and 45 degrees Celsius.
Got it.
Most human pathogens fall into this category.
Which makes sense since our body temperature is around 37 degrees.
Right.
But what about those extreme heat lovers?
Ah, yes. The thermophiles and hyperthermophiles.
Okay, so break those down for me.
Okay, so thermophiles thrive between 50 and 80 degrees Celsius.
Wow.
While hyperthermophiles can handle temperatures above 80 degrees, even boiling water.
That's incredible.
Don't let it.
Imagine bacteria casually hanging out in a hot spring. How do they even survive that?
Well, they have these special adaptations in their cell membranes and proteins that allow them to function at these extreme temperatures. Their enzymes are more heat stable, and their cell membranes have a different composition that helps them maintain their structure in those scorching conditions.
It's amazing how life finds a way to adapt, even to the harshest environments.
Absolutely.
So we've covered oxygen, pH, and temperature.
Yeah.
What other environmental factors come into play?
Well, salt concentration is a big one.
Okay.
Helophiles need high salt concentrations to grow, like the bacteria found in salt lakes.
Interesting.
Then there are halotolerant bacteria, which can tolerate salty environments but don't necessarily require them.
Got it.
And of course, light is essential for photosynthetic bacteria, which use light energy to produce their own food, just like plants.
Right.
And finally, most bacteria need moisture to grow. Water is essential for all life, after all.
That makes sense. Now, what about biofilms?
Ah, yes. Microbial cities.
Yeah, I'm fascinated by these complex communities. What exactly are they?
So biofilms are essentially groups of bacteria that stick together on a surface, encased in a slimy matrix called the extracellular polymeric substance, or EPS.
So it's like they build their own little apartment complex, complete with a protective coating.
A very apt description.
And within this biofilm, there are channels that allow for nutrient flow. right?
Precisely. It's a highly organized structure with different bacteria specializing in different tasks, much like a city.
And they communicate with each other too, right?
They do.
I've heard about this phenomenon called quorum sensing.
Yes. Quorum sensing is a remarkable process where bacteria communicate using signaling molecules.
Okay.
As the bacterial population density increases, so does the concentration of these signaling molecules.
So it's like a microbial town hall meeting where they decide what to do as a community.
A great way to think about it. Through quorum sensing, bacteria can coordinate activities like biofilm formation, virulence factor production, and even bioluminescence.
Oh,
that eerie glow you sometimes see in the ocean.
Yeah, that's so cool. And these biofilms, they can be found everywhere, right?
Absolutely. They're in nature, on rocks, in streams, on plant roots, and even inside our bodies.
Oh, right.
Think of the plaque on your teeth that's a biofilm.
Ugh. Okay, not my favorite example.
Right.
But they can be beneficial too, right?
Certainly. Biofilms play important roles in nutrient cycling and wastewater treatment.
Oh, right.
However, they can also cause problems, especially in healthcare settings.
Yeah, I was just going to say, I've heard that biofilms can make infections much harder to treat.
That's right. That's because the EPS matrix can actually protect bacteria from antibiotics and the host's immune system.
I see.
So treating biofilm infections often requires higher doses of antibiotics or even physical removal of the biofilm.
Got it.
Okay.
So we've talked about how these bacteria grow naturally.
Right.
Let's shift gears a bit and talk about how scientists grow bacteria in the lab.
Okay.
What's on the menu for these tiny creatures?
So scientists use different types of culture media, essentially bacterial food, to grow bacteria. One type is chemically defined media, where we know the exact composition of every ingredient.
So it's like a carefully crafted recipe for bacteria.
Exactly. Then there's selective media, which contains ingredients that favor the growth of specific bacteria while inhibiting others.
Got it.
This is useful for isolating particular types of bacteria from a mixed sample.
So like a VIP club for certain bacteria.
A good analogy. We also have enriched media, which contains extra nutrients to help fastidious bacteria grow. These bacteria are picky eaters and need specific nutrients to thrive.
Like a gourmet buffet for those discerning bacteria.
Precisely. And finally, there's differential media, which allows us to distinguish between different types of bacteria based on how they grow or change the media's appearance.
So it's like a color-coded system for identifying bacteria. Wow. We've covered a lot of ground here, from the basics of bacterial growth to the complex world of biofilms.
We have.
And the different media used to grow these tiny but mighty organisms.
It's fascinating stuff, isn't it?
It really is.
And now, to really test your understanding of bacterial growth, I've prepared a few questions for you based on what we've discussed.
Oh, a pop quiz. Bring it on.
All right, first question. Which of the following methods would be used to measure the concentration of bacterial contamination in processed peanut butter?
Okay, give me the options.
A, turbidity measurement. B, total plate count. C, dry weight measurement. D, direct counting of bacteria on a calibrated slide under a microscope.
This is a great question to really test my knowledge.
Right.
Let me think it through. Okay, so to measure bacterial contamination in peanut butter, turbidity is useful for liquid cultures, but peanut butter is solid, so we can rule that out.
Right.
Dry weight measurement would give us the total mass, not just the bacteria. And direct counting under a microscope would be incredibly tedious, and you might not even be able to tell the difference between live and dead bacteria.
Yeah, that's true.
So the best answer must be B, total plate count.
That's right.
This method, you're spreading a diluted sample of peanut butter on a nutrient agar plate, allowing individual bacteria to grow into visible colonies, which we can then count.
Excellent reasoning. You've clearly grasped the key differences between these bacterial counting methods.
Thanks. Okay. Feeling pretty confident now. What's the next challenge?
All right, let's test your knowledge of binary fission. What is the function of the Z ring in this process?
Okay, the Z ring. Oh, rings a bell.
Here are your options. A, it controls the replication of DNA. B, it forms a contractile ring at the septum. C, it separates the newly synthesized DNA molecules. D, it mediates the addition of new peptidoglycan subunits.
This takes me back to those mesmerizing microscopic images of bacteria dividing.
Right.
If I remember correctly, the Z ring acts like a drawstring, pinching the cell in two.
Yeah.
So I'm going with B. It forms a contractile ring at the septum.
You have a great visual memory. That's exactly right. The Z ring is a key player in binary fission, constricting the cell membrane and ultimately separating the two daughter cells.
All right. Bring on the next question.
Let's try a bit of math. If a culture starts with 50 cells, how many cells will be present after 5 generations with no cell death?
Okay, time to put my multiplication skills to the test. Each generation doubles the population, so 5 generations means doubling 5 times.
Right.
Let's see, 50 cells become 100, then 200, 400, 800, and finally 1,600 cells. So the answer is 1,600.
Perfect. You've calculated the bacterial growth spurt with precision.
Okay, feeling like a microbial math whiz now. What's next?
Let's get back to some microbiology trivia. Filamentous cyanobacteria, those fascinating photosynthetic bacteria, often divide by which of the following? A, budding, B, mitosis, C, fragmentation, D, formation of endospores.
Now, I remember those cyanobacteria forming long filaments, almost like chains. And considering those long filaments could easily break apart to form new individuals, I'm going with C, fragmentation.
Excellent deduction. Fragmentation is indeed a common mode of reproduction for filamentous organisms like cyanobacteria. You're on a roll.
Okay, bring on those biofilms.
All right, let's see if you can navigate the intricacies of biofilm life. Why is antimicrobial resistance often higher in a biofilm than in free-floating bacterial cells?
This is a critical question, especially in the context of healthcare-associated infections.
Here are the options. A, the EPS allows faster diffusion of chemicals in the biofilm. B cells are more metabolically active at the base of a biofilm. C cells are metabolically inactive at the base of a biofilm. D, the structure of a biofilm favors the survival of antibiotic-resistant cells.
Okay, if those cells at the base of a biofilm are not actively growing and dividing, then antibiotics targeting those processes might be less effective. So I'm leaning towards C.
That's a sharp observation. You've identified one of the key reasons for antibiotic resistance in biofilms. That slower metabolic activity in the deeper layers of the biofilm makes those bacteria less susceptible to many antibiotics.
Interesting.
Additionally, the EPS matrix can act as a physical barrier, hindering antibiotic penetration.
Right.
And you're right, the structure of a biofilm can indeed favor the survival of antibiotic-resistant cells, so option D also plays a role.
So it's a combination of factors that make biofilms such a challenge to treat.
Absolutely.
This really highlights the importance of understanding microbial growth in all its complexities.
Absolutely. It's not just about individual cells.
Right.
It's about how they interact and adapt within these complex communities.
Okay, ready for another round of biofilm brain teasers.
All right, let's delve deeper into bacterial communication. Quorum sensing is used by bacterial cells to determine which of the following: A, the size of the population; B, the availability of nutrients; C, the speed of water flow; D, the density of the population.
Quorum sensing. It's all about bacteria sensing how many of their buddies are around, right?
Right.
But is it the size of the population or the density? They seem similar.
You hit on a subtle but important distinction. While quorum sensing is related to population size, the key factor is the density of the population. It's about how closely packed the bacteria are.
I see.
Which influences the concentration of those signaling molecules they use to communicate.
So it's not just about how many there are,
right,
but how crowded they are.
Exactly.
Well, that makes sense. The more crowded, the stronger the signal,
right,
and the more coordinated their actions become.
Precisely. You're really starting to think like a bacterium now.
Okay, one more question before we move on to exploring bacterial growth in different environments.
All right, last one for this round. Which of the following statements about autoinducers, those signaling molecules involved in quorum sensing, is incorrect?
Okay, hit me with the options.
A, they bind directly to DNA to activate transcription. B, they can activate the cell that secreted them. C, enacylated homoserine lactones are autoinducers in gram-negative cells. D, autoinducers may stimulate the production of virulence factors.
Option B sounds familiar. Bacteria can respond to their own signals. Option C seems like a specific detail I vaguely recall.
Okay.
And option D makes sense. Virulence factors are often regulated by quorum sensing.
Right.
So the odd one out is A, they bind directly to DNA to activate transcription.
Yeah.
That sounds more like the role of transcription factors, not these signaling molecules.
You've nailed it again. Autoinducers don't directly bind to DNA.
Yeah.
Instead, they bind to specific receptors, which then trigger a cascade of events that can lead to changes in gene expression.
Got it. So now that we've covered the basics of bacterial growth and reproduction, as well as their fascinating communication systems, let's explore how these microbes thrive in different environments, from those that love oxygen to those that avoid it like the plague. Okay, so let's dive into those different bacterial lifestyles based on their oxygen preferences. It's amazing how some bacteria thrive in oxygen-rich environments, while others can't even tolerate a whiff of it.
It really highlights the diversity of metabolic strategies that have evolved in the microbial world. Let's start with a classic microbiology experiment using thioglycolate medium, which is designed to test oxygen tolerance. Imagine an inoculated thioglycolate medium culture tube that shows dense growth at the surface and turbidity throughout the rest of the tube. What would you conclude about the oxygen preferences of those bacteria?
Well, we're seeing growth throughout the tube, which means the organisms can tolerate oxygen to some extent, but that dense growth at the surface suggests they really prefer oxygen. So this points to facultative anaerobes, right?
Exactly. Facultative anaerobes are the multitaskers of the microbial world, able to switch between aerobic and anaerobic respiration depending on the oxygen availability.
They're flexible like that.
Now, let's flip the scenario. What if the inoculated thioglycolate medium culture tube is clear throughout the tube, except for dense growth at the bottom of the tube?
Well, that's a clear indication that those organisms are avoiding oxygen. They're only growing at the bottom where oxygen is minimal, which suggests they can't tolerate it. So we're dealing with obligate anaerobes, those that rely on anaerobic respiration or fermentation, and can't survive in the presence of oxygen.
You've decoded those experimental results perfectly. Now let's apply this knowledge to a real-world example. Pseudomonas aeruginosa is a common pathogen that infects the airways of patients with cystic fibrosis. It's known that it doesn't grow in the absence of oxygen. Based on this information, what type of bacteria would you say Pseudomonas aeruginosa is?
Let's see. Pseudomonas aeruginosa infects airways where oxygen is plentiful, and it can't grow without oxygen, so that clearly points to an obligate aerobe.
Spot on. Now let's move on to another bacterial culprit, this time one that's infamous for causing cavities. Streptococcus mutans resides in the gum pockets, lacks catalase activity, and can be grown outside of an anaerobic chamber. What can you deduce about its oxygen preferences?
This one requires a bit more thought. Streptococcus mutans lives in gum pockets, which have limited oxygen. And the lack of catalase activity suggests it can't handle high oxygen levels. But it can grow outside an anaerobic chamber, so it's not a strict anaerobe. So I'm thinking it must be an aerotolerant anaerobe.
Brilliant deduction. Aerotolerant anaerobes have their own ways of dealing with oxygen's toxic effects, even without catalase.
It's amazing how they've adapted.
Now, shifting gears a bit, let's talk about those bacteria that thrive in environments with higher than normal carbon dioxide levels. Why do you think the instructions for growing Neisseria gonorrhea, the bacterium responsible for gonorrhea, recommend a CO2-enriched atmosphere?
Okay, so Neisseria gonorrhea needs extra CO2, but it's not using it for respiration or photosynthesis. And it's not a strict anaerobe. So the answer must be that it's a capnophile. One of those bacteria that has a special affinity for carbon dioxide and grows best in its presence.
That's exactly right. Some capnophiles, like Neisseria gonorrhea, require higher CO2 levels for certain enzymatic reactions to occur properly.
Fascinating. Okay, what about those bacteria that thrive in extreme pH conditions?
Yes, the acidophiles and alkalophiles. They've evolved to handle environments that would be toxic to most other organisms. Think about bacteria that grow in mine drainage, where the pH can be as low as 1 or 2. What category would they fall into?
Mine drainage is highly acidic, so those bacteria must be acidophiles.
Yes.
The ones that thrive in acidic environments and have special mechanisms to maintain a stable internal pH.
Precisely. Now, for a contrasting example, consider the bacteria that live in Lake Natron, where the water pH is close to 10. What type of pH extremophiles are those?
Lake Natron with a pH of 10, that's incredibly alkaline. So we're looking at alkalophiles, the ones that thrive in those alkaline environments, and often use sodium ions to drive their metabolic processes.
You've got it. Alkalophiles often have unique enzymes and cell membrane adaptations to cope with those high pH levels. Speaking of extreme environments, let's shift our focus from pH to temperature. Bacteria have truly conquered a remarkable range of temperatures.
Right, from the icy cold to the boiling hot. They've adapted to it all.
Indeed. Let's start with those sneaky food spoilers. Imagine a soup container that was forgotten in the refrigerator and now shows contamination. What type of bacteria are most likely the culprits?
Okay, let's see. Thermophiles love heat, so they're out. Acidophiles prefer acidic conditions, not the neutral environment of soup. And while mesophiles grow best at moderate temperatures, which might include room temperature, the refrigerator is much colder. So the answer must be psychotrophs, right?
Spot on. Psychotrophs are a major concern in food safety because they can thrive in refrigerated conditions and cause food spoilage, even if the food appears to be properly stored.
Makes sense.
Now let's turn up the heat a bit. What type of bacteria would you expect to find thriving in a hot tub set at a comfortable 39 degrees?
Well, 39 degrees Celsius is warm, but not extreme enough for thermophiles or hyperthermophiles. And psychotrophs prefer colder temperatures.
Yeah.
So the most likely inhabitants of a hot tub are mesophiles.
Exactly. Mesophiles are the most common type of bacteria we encounter in our daily lives as they thrive in temperatures that are comfortable for us. Now, let's crank up the heat even further. Where would you expect to find a hyperthermophile, one of those extreme heat lovers?
We're talking extreme heat. A hot tub and warm ocean water are too mild for hyperthermophiles. And the human body is definitely out of their comfort zone. That leaves us with a hydrothermal vent at the bottom of the ocean.
You nailed it. Hyperthermophiles have special enzymes and cell membrane adaptations that allow them to function at temperatures that would literally cook most other life forms.
That's incredible. Okay, to round out our temperature tour, let's consider those cold-loving champions, the psychophiles. Which of the following environments would be their ideal habitat? A contaminated petri dish left in a 35°C incubator, yogurt cultured at room temperature, a salt pond in the desert with a daytime temperature of 34°C, or a mountain lake with a water temperature of 12°C?
Good question.
Well, an incubator and a desert salt pond are way too warm for those cold-loving psychophiles, and yogurt is cultured at a moderate temperature. That leaves us with the mountain lake.
Excellent. It's amazing how diverse the microbial world is, with specialists adapted to all sorts of extreme temperatures. Now let's talk about another environmental factor that can affect bacterial growth: water activity. Some bacteria are surprisingly tolerant of dry conditions.
Right. Water activity is essentially a measure of how much water is available for microbial growth.
Exactly. Think about jams and dried meats. Why do you think these foods often don't require refrigeration to prevent spoilage?
Jams are acidic, which can inhibit some bacteria, but not all. And dried meats don't usually have toxic alkaline chemicals or naturally occurring antibiotics. So the key factor must be their low water activity, right?
Precisely. Reducing water activity is a simple but effective way to inhibit microbial growth, and that's why drying and preserving techniques have been used for centuries to extend the shelf life of food.
It's fascinating how humans figured that out.
Speaking of harsh environments, let's talk about those bacteria that thrive in salty environments.
The halophiles. Those salt lovers are truly remarkable.
Indeed. Imagine a salt marsh with its high salt concentrations. What type of bacteria would you expect to find thriving there?
Salt marshes are definitely not the place for acidophiles, barophiles, or thermophiles. So the most likely inhabitants are halotolerant bacteria.
Absolutely. Halotolerant bacteria are often found in environments where salt concentrations fluctuate, like estuaries and salt marshes. Now, shifting back to the lab, let's talk about the different types of culture media used to grow bacteria.
Right. Scientists have a whole arsenal of bacterial food to choose from, depending on what they're trying to grow.
Here's a question about one of those specialized media. agar is used to identify and isolate pathogenic bacteria. It contains digested meat proteins for nutrients and two indicator dyes, eosin and methylene blue, which inhibit gram-positive bacteria and distinguish between lactose-fermenting and non-lactose fermenting organisms. Lactose fermenters form metallic green or deep purple colonies, whereas non-lactose fermenters form colorless colonies. Based on this information, what type of medium would you say agar is?
Let's see. It contains digested meat proteins, which means it's a complex medium, not chemically defined. It inhibits gram-positive bacteria, making it selective, and it differentiates between lactose fermenters and non-lactose fermenters, making it a differential medium. So the answer is that agar is a selective, differential, and complex medium.
You've analyzed that perfectly. agar is a powerful tool in microbiology, allowing scientists to select for specific bacteria and differentiate them based on their metabolic capabilities. Now, for our final question, let's consider Haemophilus influenza, a bacterium that must be grown on chocolate agar, which is blood agar treated with heat to release growth factors. Based on this specific requirement, how would you describe Haemophilus influenza?
Well, this suggests that Haemophilus influenza is a fastidious bacterium, one that has complex nutritional requirements and needs specific nutrients to grow. It's amazing how scientists have figured out the culinary preferences of these microscopic organisms.
It is fascinating, isn't it? We've covered a lot of ground in this deep dive into the world of microbial growth, from how they reproduce, the environmental factors that influence their growth, their intricate communication systems, and the remarkable diversity of their lifestyles.
And we've even tackled some challenging questions to test our understanding of these tiny but mighty organisms.
It just goes to show that there's always more to learn about the microbial world.
So to our listeners, we leave you with this thought-provoking question. Scientists are discovering new extremophiles all the time, pushing the boundaries of where we thought life could exist. What does this tell us about the limits of life? And could there be microbes thriving in environments we haven't even imagined yet? Keep exploring, keep questioning, and keep diving deep into the fascinating world of science.